Agglutination tests. Agglutination reactions are based on the interaction of antibodies and particulate antigens in presence of electrolytes at a suitable temperature and pH. The antibodies involved in the reaction are also known as agglutinins, and particulate antigens are also known as agglutinogens. Antigen and antibodies interact to form visible large clumps. The test could be qualitative or quantitative. The amount of agglutinate formed is greatly influenced by the relative proportion of antigens and antibodies, antibody avidity, time of incubation, and the test environment. Agglutination tests can be broadly classified into direct agglutination, also known as active agglutination, indirect or passive agglutination, and co-agglutination. Direct agglutination, where the antigens are found naturally, example between a cellular antigen and its antibody. Indirect or passive agglutination, the reactions employ carrier particles that are coated with soluble antigens. Carriers can be biological or artificial. Number 3 is co-agglutination, which uses protein A. Direct agglutination test. It is the direct agglutination of cellular antigen by its corresponding antibody. When cells interact in vitro with the appropriate antibody, they clump together and eventually form masses that become large enough to be seen. Direct agglutination is used for detecting antibodies in tularemia, brucellosis, listeriosis, and rickettsial diseases. The reactions can broadly be of following types. Number 1, slide agglutination. Number 2, tube agglutination. Number 3 is heterophile agglutination, and number 4 is antiglobulin or Coombs test. Slide agglutination test. It is a qualitative agglutination test performed on a slide, utilized as herd screening test for many bacterial diseases. Commonly used for rapid identification of bacteria, used as Vidal reaction for diagnosing typhus and paratyphus. Examples include blood typing, Rose Bengal plate test, Salmonella pullorum plate test, Lensfield method for serotyping of streptococci, etc. In this test, bacterial suspension is added to the drop of standardized antiserum. Clumping of bacteria occurs within seconds indicates a positive test. Control consisting of antigen suspension in saline without adding antiserum is included on the same slide. This is Rose Bengal plate test, where the test antigen is heat inactivated Brucella abortus cell stained with Rose Bengal and buffered at pH 3.65. Place 30 microliter of test antigen next to 30 microliter of serum on a flat, white or transparent surface. Mix the drop with a stick producing a zone of approximately 2 cm diameter. Rock the slide and gently for about 4 minutes. The formation of agglutinates is the positive reaction, and no formation of agglutinates indicates negative reaction. Next is tube agglutination test. It is a standard method used for quantitative estimation of antibodies in the serum. Test is performed in glass tubes using serial dilution method. It is routinely used for serodiagnosis of enteric fever, that is Vidal test for typhoid and brucellosis. The method consists of adding a fixed volume of antigen into test tubes containing serial dilutions of antiserum in normal saline. A positive reaction is indicated by formation of visible clumps after incubation. The reciprocal of highest dilution of antiserum giving positive reaction is the titer of antibodies. Appropriate antigen and serum controls should be included to validate the results. Heterophile agglutination test. This test depends on demonstration of heterophilic antibodies in serum present in certain bacterial infections. Antiglobulin test, also known as Coombs test. Coombs test was devised originally by Coombs, Mourant and Reyes. The test is used for detection of anti-Rh antibodies, incomplete antibodies in brucellosis and other diseases. Coombs test is of two types, direct Coombs test or indirect Coombs test. When serum containing incomplete anti-Rh antibodies is mixed with Rh1 erythrocyte in the saline, incomplete antibody antiglobulin coats the surface of erythrocytes but does not cause any agglutination. When such erythrocytes are treated with antiglobulin or Coombs serum, that is rapid antiserum against human G globulin, then the cells are agglutinated. The principle is that the antibodies produced against Rh antigen are of IgG type. Number of Rh epitopes are less and sparsely placed on the surface of RBCs. The negative charge on the surface of RBCs also prevent them from coming closer than within 20 nanometer of each other. As IgG antibodies are bivalent, they are unable to cross-link Rh epitopes present on the surface of RBCs and therefore are unable to agglutinate RBCs. Therefore, the antibodies formed against Rh antigens are called incomplete antibodies. It is a misnomer as IgG antibodies are complete with bivalency, and the problem is with Rh antigen. Direct Coombs test. In this test, the sensitization of RBCs with incomplete antibodies takes place in vivo. The cell-bound antibodies can be detected by this test, in which Coombs serum is used to agglutinate patients' red cells. The occurrence of agglutination means that the patient's RBCs have cell wall-bound antibodies. Direct Coombs test is used for diagnosis of hemolytic transfusion reaction, hemolytic disease of the fetus and newborn, autoimmune hemolytic anemia, etc. Indirect Coombs test. The indirect Coombs test detects the antibodies against human RBCs in the patient's serum. In this method, the patient's serum is incubated with RBCs of a known type, followed by the addition of Coombs serum. If test serum contains incomplete circulating antibodies, they will sensitize RBCs in vitro and agglutination will occur. The indirect Coombs test is used in cross-matching before blood transfusion and in prenatal testing of pregnant women. Indirect agglutination test or passive agglutination test. It is an agglutination test where the soluble antigens are coated or adsorbed onto a carrier particle. Carriers can be biological, like for example erythrocytes, or artificial, for example latex particles. Binding of specific antibodies to such adsorbed antigens result in the agglutination reaction. When the antibody is adsorbed on the carrier particle for detection of antigens, it is called reverse passive agglutination test. Depending on the carrier particles used, the test can be of following types: latex agglutination test, heme agglutination test, and co-agglutination test. Latex agglutination test. It is a test that employs latex particles as a carrier of antigen or antibodies. In 1955, Singer and Plotz accidentally found that IgG was naturally adsorbed to the surface of polystyrene latex particles. Use of monoclonal antibodies has reduced the cross reactions, resulting in reduction of false positive reactions. There are many commercial latex particles tests available. For example, for grouping streptococci, for detection of bacterial antigens from cerebrospinal fluid, for detecting rheumatoid factors, for detecting rotaviruses and adenoviruses from the stools, etc. Latex particles are inexpensive, relatively stable, and are not subject to cross-reactivity with other antibodies. Additionally, the large particle size of the latex facilitates better visualization of antigen-antibody reaction by the naked eye. The tests are usually performed on cardboard cards or glass slides, and positive reactions are graded on the scale of 11 to 41. Direct hemagglutination test. Some viruses contain hemagglutinins that can directly bind and agglutinate mammalian and avian red cells. Hemagglutination is an agglutination of red blood cells. Hemagglutinating viruses include automyxoviruses, paramyxoviruses, flaviviruses, and bunyaviruses, as well as some adenoviruses, rheoviruses, parvoviruses, and coronaviruses. Direct hemagglutination is also used for diagnosing of infectious mononucleosis, for detection of cold agglutinins in pneumonia caused by Mycoplasma pneumoniae. Reverse passive hemagglutination. When antibodies are attached to the RBCs to detect microbial antigen, it is known as reverse passive hemagglutination. The test has been used extensively in the past to detect viral antigens such as HBs antigen in the serum for diagnosis of hepatitis B infection. Hemagglutination inhibition test. Some viruses agglutinates red blood cells. The process of hemagglutination can be inhibited by antibodies specifically directed against the virus, and this phenomena is called hemagglutination inhibition. To perform this test, patient's serum is first incubated with a viral preparation, then RBCs susceptible to the virus are added to the mixture. Absence or reduction of agglutination indicates the presence of antibody in patient's serum. Indirect or passive hemagglutination test. When RBCs are coated with the antigen to detect the antibodies in the serum, the test is called indirect hemagglutination test. Red blood cells which are first treated with ethanic acid or formalin are carriers of soluble antigens. Antigen is either adsorbed or bound to the erythrocyte surface. Used to detect antibodies against Enterobacteria, Clostridium tetani, Treponella pallidum, and against some tissue antigens like thyroid gland antigens. The test involved dilution of test sera followed by the addition of an equal volume of virus suspension diluted to contain approximately 4 to 8 HA units. The appropriate RBC suspension is then added. Plates are gently mixed and allowed to incubate for 1 to 2 hours. If the specific antibody is present in the test sera, hemagglutination will be inhibited and RBCs will settle as well-defined pattern. Agglutinated cells, in contrast, will settle out in a thin layer over the entire bottom of the test well. Co-agglutination test. Co-agglutination is a type of agglutination reaction in which CoA1 strain of Staphylococcus aureus is used as carrier particle to coat antibodies. CoA1 strain contains protein A, an anti-antibody that combines with the Fc portion of immunoglobulin G, leaving the FAB region free to react with the antigen present in the specimens. In a positive test, these coated antibodies will be agglutinated if mixed with specific antigen. Advantage of the test is that these particles show greater stability than latex particles. This reaction is used for identifying serological types of Streptococcus species, Neisseria meningitidis, and hemophilic influenza. Thank you for watching. Don't forget to subscribe for more such videos.